So good afternoon. Thanks the leadership group for letting me to give talk here. I come from Tianjin University. Basically, I'm in engineering school. Why we are interested in IC 2.0 project? So most case we in the application side try to produce some medicine and biofuel, try to track the problem arising from environmental or some biodetections. For us, we want a designer cell. So the designer cell, by have a designer cell, we can improve the existing system, the efficiency, and even create completely new applications. That's why we are interested in the SC 2.0 project. And the key issues for the implication of designer cell, when we want to get a gain of function or to produce a product, we design and build a functional model. We insert a wild-type microbiome. Most cases in these systems cannot be work very well. So what we're trying to do is that both ways. One is optimize the functional model, another is try to engineering the chassis. That's why we have so great interest in this project. That's by optimize this and re-engineering, redesign, and reconstruction of this chassis, it can be get a perfect systems. So that's why we have spent so much effort in this project. So for doing so, I would like to introduce this side by designing chemical synthesis, assembling, debugging, and scrambling systems. So TJU, as a core member of IC 2.0, is responsible for chemically synthesis of chromosome 5 and chromosome 10. And here shows the features of this design. For chromosome 5, we have 176 log P side. This may be used for chromosome. And we also have 339 P side tags also can be used for. Okay. Also, for chromosome 10, we have a very similar design. So what we learned from doing this project, that's for building a designer chromosome. We learned that firstly we design that and build, test, and bug mapping and debugging and learn knowledge, and then we can redesign. So it is a circle. By maybe several rounds of this cycle, we can get a perfect chromosomes. For the chromosome 6, it was incorporation by 17 stepwise of incorporations. So that means for each of this large mega trunk, we can divide it into the mini trunk and then divide more small building block. So that's we can parallelly build this the 17th mega tracks. That's we introduce another course that's called BAJ, originally in Johns Hopkins by Joe's lab. So we introduce this build a genome in TJU. And yeah, we have about each year about 120 students take this course, and then some of them take the experiment. By take this experiment, we try to using these teams to form different teams. Each day, the team inside the team they have a lot of discussion, and for a week, different group, different teams are working together, and by brainstorm teamwork, they have improved the technical a lot. From this timeline, you can see for this chromosome, for this since chromosome 5, it takes much longer than the draft because this the start for chromosome it started 2011 at the month of 6, but this time it takes much shorter time. By comparisons, to get a draft version, it takes for a month. We can chemically build about 23 kilobase, but for draft 10, we have much more fast speed because we learn a lot. And the next is the test, and we test the genotype, phenotype, computer growth, and stability, 3D organization, and omics analysis, and the phenotype. All this should be near the wild type phenotype, so we get a better one. If there's something wrong, it may be a defect, growth, or laser, then we should bug mapping. So we develop a bug mapping called POM, and develop several types of different types of bugs. And for chromosome 5, after the build up, for deep sequencing, we found 34 variants. We're using about 9 months to correct all of these variants to get a perfect designer chromosome. By this correction, we're using the integrative core transformation method, CRISPR-mediated, and the combination of both these. And during the repair process, in some cases, for each of the round of corrections, we can get 3 the variant be corrected. Sometimes get 2, so it's pretty efficient. And finally, we by about 22nd round of corrections, we all of the 34 variants were corrected to the designer sequencing with another 2 new variant. So we spend more time to repair all of the. And next, I would like to introduce the potential applications of this designer chromosome. With up to now, 1/3 of the chromosome were designed and chemically synthesized. Because this is a scramble synthesis as introduced by Professor Dai, so it can produce a lot of different combinations. So originally, using a non-designer chromosome, we only take one way or another. But because we have this so many log-speed side within each chromosomes, we can have beyond the pathways the DNA segment arrangement. So we may have more choice. So for scramble within the designer is such for chromosome 5 we have 176 log-speed side. So about if we have induced the query, it can lead to the deletion, inversion, relocation, duplication, as Professor Dai said. So by doing this, in most cases there's some leakage. We designed different. We designed 4 types of the switch. By comparison, we found this one is a favorite one, and then we form an AND gate. By testing the AND gate, we found it's very tight control the process. And for the first round of scrambling the synth3 and synth5 diploid yeast, we found that it can, by first run, it increase the production dramatically. So when we deep sequencing, we found that there are larger DNA segment. It's about 17.6 kilobase DNA fragment deletions observed during the first round of scramble. It contains 2 essential genes and 7 non-essential genes. So we get a lot of improvement. So next, we make a multi-scrambling improvement second. That's we scramble them, get the diploid, and then grows again, and re-scramble them. And up to now, we have 5 rounds of this 5 rounds of this scrambles. It increased about 70 37-fold increase of this production. And we also calculated the frequency about the some cases you can have the it becomes darker. The rate become darker is roughly around about 10%. And this said, can you have a method to have optimize the functional models? So in fact, we need different combinations with this model. So we developed so-called in vitro scramble systems. By construction in vitro, induce the create in combination, we can have different pools, and by in vivo type phenotype sequencing, we can get a different combination of these models and different productivity. And the next slide shows that the correlation of phenotype with the genotype by this in vitro scramble synthesis. So it's very powerful. So generally, we have into the stage of developing applications for ISO 2.0. And also, we think the BAJ China course is successful combination research with education. So we get training a lot of manpower and optimize technology. So it can be used in current project, genome project, right? And both our technology are very useful for many new technology. Thanks collaborators and the group members. Thank you.
Hi. Over here. Hello. Hi. So for entropic reasons, typically chrrecombinase causes deletion because eventually you get smaller and smaller fragments, so you have just one lock site. So do you really get, when you sequence these clones, do you really get a diversity of modifications, or are you mainly getting deletion of genomic fragments?
And this talk I only shown one case. That's a larger DNA fragment. In fact, we are deep sequencing about 18 strains we got. So maybe later we show you more some deletion, duplication, or the case.
Just to add to that, in our previous study, we showed that the frequency of deletion and inversion is roughly equivalent. And one of the big benefits of doing this in a diploid is that deletions don't have as big an impact. Obviously, you can delete an essential gene if you're in heterozygous diploid, so you preserve a lot more of the diversity.